Thank you for this nice summit. And before
can you make it full screen, Professor?
Yes, sure.
Excellent, good to go.
The first thing about nutrition is that we need commodities and we need to have available food for all the population. So the main problem in agriculture are plant disease that treat crop yield and food security. And pathogens evolve their genomes and produce new effector which can overcome resistance genes. So today we have a good variety that are resistant to their pathogens, but this resistance lasts for a short period. So evolution brings us new strains that are more invasive, and we have now available modern technologies that make improving resistance genes or introduce several resistant genes at the same time. But most related to this presentation is the understanding of the immune defense mechanism and signaling to protect crops from pathogens. Shortly, I will briefly describe what is the mechanism of plant immunity. There is at first recognition in the apoplast between the cell wall and the membrane of molecular patterns, and these receptors make the first signaling of danger. So following this step, there are the injection in the cytosol of the pathogen effectors, and the plant senses this effector with the resistance genes and activate effector trigger immunity. And this proposed model was named the Zig model. But today we do not consider these events independently. And this is a circular process in which effector trigger immunity cooperates with pattern trigger immunity. And we have also the availability of resistance genes and in wild species. So, for example, in wheat, we have 4 genes that can be transferred to barley and confer resistance to stem rust. But we need always new solutions, because this effector evolution is evolving. So we proposed recently the swap of leucine-rich repeat elements to receptors, and we showed that these chimeras confer more enhanced resistance in Solanaceae. But again, there are many other possible interventions. For example, in Cladosporium fulvum, avirulence effector AVR9, it is sensed by a receptor that's called CF9 in tomato. And this recognition of effector is given by this localized hypervariable region in the 4 central using rich repeats. So what I researched in the last years was the transfer of this region that recognize one ligand to resistant genes and receptors, and we showed that these chimeras or swapped resistant genes are effective to increase resistance. But coming to the talk of this presentation, nicotinamide adenine dinucleotide is a substrate of enzymes. It is required for ADP ribosylation. It is required for citrulline, that the acetylate proteins, and is required for TIR domain. It is stay for tall interleukin receptor domain. This domain is present in resistant genes, and TIR domain hydrolyze NAD to form nicotinamide and cyclic ADP ribose. Coming to the ADP ribosylation reactions, these reactions are reversible, so require enzyme writers and enzyme erasers. In this case, the poly-ribose polymerase is activated by DNA strand breaks, auto-modified itself, and this is the PAR cycling. We can see the schistons of the glycohydrolase that reduce the polymers, and this PAR cycling consumes most of the NAD in the cell, and this NAD depletion is linked to cell death. But we are interested in plants, so we take only the main meaning of this reaction. So we have writers, erasers, and MAR or PAR readers. These are protein with domain that recognize ADP ribose modifications and are involved in forming protein complexes. In plants, we have 2 glycohydrolase genes, 3 PARP genes, but also we have PARP domain proteins that I will show you what is their function. This is slide and the next show how the PAR readers recognize modified proteins, and for this presence specifically bind to these domains, and once attached have the function to form protein complexes. And in the kingdom of life, we have several about 250 PARP sequences, and they are grouped for their homology. In group 1, we have human PARP 1, PARP 2, PARP 3, and plant PARP 1 and PARP 3 that contain zinc fingers, and PARP 2 that contain another domain that bind nucleic acid. And this work by Pater and Rissel showed that when we mutate the three parts in Arabidopsis, we don't see any difference. They studied accumulation of callose in the cell wall, and when plant is challenged with the flagellin 22 peptide, there is still deposition of callose, but only when they use the PARP inhibitors, this immune response was blocked. So pyrylation even increased in mutant. So there are other proteins that are responsible for this activity, and these are PARP domain proteins. And these are two types of part domain: the radical cell depth 1 and similar to radical cell depth 1. They also studied the mutants, and they showed that is reduced growth, there is stress like response, and the plants are resistant to such stress and to reactive oxygen species. And this schema show that radical cell 1 has the part domain which bind nicotinamide adenine dinucleotide, has a glutamate tryptophan domain that interact with other proteins, and there is a RST domain that interact with transcription factors. So when we produce ADP ribose units and cleave these ADP ribose units, we have an accumulation of ADP ribose that is processed by these Nudix hydrolases to produce adenylase monophosphate and ribose 5-phosphate. And all these small compounds are recycled and reconverted. So this is the Nad Salvage Pathway that is started from nicotinamide. This is transformed in a nicotinamide mononucleotide, and this reforms NAD. And these Nudix hydrolases are also involved in plants to interact with the salicylic acid immune defense mechanism. So these are all these intermediate products are important for the defense of plant and immunity. Coming to the toll interleukin-1 receptor domain, TIR domain, it was shown that either in animals and in plants, TIR domain oligomerize and cleave NAD. So when NAD is cleaved by TIR domain, the signaling molecule cyclic ADP ribose is formed. In human, the main protein with interest in this activity is the NADase called steroid alpha and toll interleukin receptor motif containing 1, SARM1. So it is interesting that SARM1 is activated by nicotinamide mononucleotide, and this not happens in other NADases. So when the permeable mimetic CZ48 was used, it was shown that there is accumulation of cyclic ADP ribose in vitro and in vivo. And this is the localization of the NADases in humans. CD38 is non-TIR domain NADases that faces the extracellular space and also the intracellular space, while SARM1 is localized to mitochondrial membrane and faces the cytosol. And this is where the cyclic ADP ribose production occurs. And this is just a talk about the other kingdom of life, and this is plants again. So the TIR domain in plants can form modular proteins like TIR domain alone, TIR domain associated with the X domain, TIR domain with the nucleotide binding site leucine rich repeat region that is typical of resistant proteins, and other possibility to association. So this is the variant cyclic ADP ribose that is found in plants, and its structure is still not determined. So what happens downstream? So, this activation of cyclic ADP, two helper resistant genes. One is enhanced disease susceptibility 1, and the other RPW8. And these this involvement produce formation of two types of resistosomes. So it is important to know that when we pre-treat Arabidopsis plant with nicotinamide mononucleotide, it shows increased resistance to Fusarium graminearum. And this is, again, the formation of the two types of resistomes that activate immune defense. So now we know that we need a high level of NAD for all these reactions. So NMN and NAD must be available for these enzymes. And NAD is consumed, so we need efficient Nad salvage pathway and functional to regulate NMN levels. And this is in the schematic slide of NAD salvage pathway in humans. We can see there are three compartments of NAD. Nuclear, mitochondrial is cytosolic. The enzyme of the Nad salvage pathway are NAD mononucleotide nicotinamide phosphoriboside transferase that produce NMN, and nicotinamide mononucleotide adenine transferases. There are three types. One is nuclear and is not a reversible reaction, so we start from NMN and produce NAD. The other two are cytosolic and mitochondrial, and these reactions are reversible. Then one enzyme important on which we can play to activate and increase this NAD synthesis is NMNAT. So we can stimulate an NMNAT, or we can induce the expression of NMNAT, and when NMNAT1 is knocked down, we found elevated levels of NMN that activates cyclic ADP ribose production. So the NAD pools are beneficial for plant immunity, since NAD stimulates salicylic acid-mediated signaling and pathogen resistance. And it is possible to sustain NAD levels and increase plant immunity. It was shown that exogenous application of NAD or NMN protects barley from powdery mildew, and in Arabidopsis protects from Fusarium graminearum and Pseudomonas syringae. Therefore, it is possible to deliver through exosomal liposomes NMN to provide plant cells with NAD precursors. Conclusion. So to overcome bacterial and fungal disease, we have engineering technologies, but also we have the possibility to deliver exosomes or liposomes to supplement cells with nicotinamide mononucleotide or NAD. And this is the acknowledgement to my collaborators, Alexander Brutus, Barbara Reca, and Fedra Franclocci, with our publication on this topic. Thank you for your attention.